time. I guess we'll just go ahead and get started. It's 3:01. Good afternoon, everyone. Thanks for those who are showed up in person here, the new faces I've met, and those who've joined through Zoom. For those who I haven't had the pleasure to meet yet, my name is Justin Joy. I'm our outpatient clinical pharmacy specialist, part of the specialty and ambulatory care team at Emory University Midtown. And today, my grand rounds will be good data gone bad. Has evidence-based medicine really been hijacked? For the sake of time, I'm not going to read through all of my objectives, but we're basically going to go over three things. We're going to go over what evidence-based medicine is. We're going to talk about some of the current challenges and debates in evidence-based medicine. And lastly, what are some potential strategies on how to deal with some of these challenges and limitations? I've got my disclosures up there for reference for you guys to read. And this is going to be pretty ironic later. So just keep that in mind. And secondly, I want to disclose the fact that I am a very strong advocate of evidence-based medicine. I think some of my learners and colleagues would say I might be the #1 proponent of evidence-based medicine. But I do feel like if we can at least acknowledge some of the negative sides of evidence-based medicine, then we can't really call ourselves true proponents. And I don't think any of the information will necessarily be controversial, but I do want to leave it up there just in case. And I also do want to say that I am still learning, and I am no expert in medical evidence. I am not a biostatistician, and I am no expert trialist by any means. But I want to start off with a simple question, not necessarily a learning assessment, but who would agree that pharmacy and medicine in general is hard? It's not easy. Any falses? Anyone think it's false? Good. Yeah, it is very, very hard. And it's something that I've definitely realized. In a quick backstory about myself. So I did my PGY1 training actually right here at Emory University Hospital. And I gave my grand rounds at this exact podium 5 years ago. And everything still looks pretty much the same. And I did my PGY2 in cardiology at Duke University Hospital. And one of the things I loved about cardiology was it is loaded with medical evidence, and we spent quite a bit of time on literature evaluation and critical appraisal. And what I loved about Duke in cardiology was that it really was where all the excitement was happening. A lot of the principal investigators who I only knew through seeing their names on paper were then right there in front of me on rounds, and I got a chance to pick their brains. And so when I left residency and I started my outpatient position here at Emory, I really thought I had a pretty good grasp of it. But then I realized that the more you see, the harder it gets, and that really is true. And I want to share you guys a story from the last story, actually, that one of my preceptors shared with me right before I left for my graduation of PGY2. And that story is about the little pill that could. And this story dates back to 1977, where cardiologists noticed these PVCs, which are premature ventricular contractions. And these are basically little blips that you can see on an EKG after someone has had a myocardial infarction. But what was concerning was that there was a direct correlation between these PVCs and all-cause death. And this was a major problem, and this was seen as signs of doom. And Bernard Lawn, who's actually the inventor of the defibrillator, gave a talk to the American College of Cardiology and wrote a paper on how this is being one of the biggest challenges confronting modern cardiology. And he advocated that we needed some antiarrhythmic drug to help prevent these PVCs and ultimately patients going into ventricular fibrillation. Well, lucky for him, not too long after that, 2 years later, we got a drug called flecainide. And this paper was published in the New England Journal of Medicine. And flecainide was amazing at treating these PVCs after MIs. And so in CCUs across the country, there are stories that I hear about on videos and in papers of cardiologists talking about how this was the standard of care. In all CCUs across the country, the use of flecainide post-MI for PVCs was typical. But there wasn't actually an adequate randomized controlled trial looking at clinical outcomes. And one of the issues was people thought it was unethical to even run such a study. These PVCs were directly correlated with death. How can you let patients just sit by and have these PVCs go untreated? But luckily, there were some really brave investigators who started the CAST trial. And the first part of this study was published in 1991. And the CAST trial stood for the Cardiac Arrhythmia Suppression Trial. And the CAST trial took about 2,000 patients who had a myocardial infarction or heart attack and also had these PVCs. And they were randomized to either an antiarrhythmic, which was flecainide or its close cousin echinide, or placebo. And their primary endpoint was death or cardiac arrest due to arrhythmia. Now, does anyone want to guess what the results of this study were? Any guesses, any takers, nothing? Well, I know it surprised me, even looking back in hindsight, but there was actually an increase in mortality in the antiarrhythmic group. So 8.3% in the antiarrhythmic group versus 3.5% in the placebo group. And this was basically a number needed to kill of 21 over the course of 10 months. Something that was the standard of care and no one even thought twice about. And this was, very concerning, and it really is a cautionary tale of one that sometimes it is important to challenge the standard of care if there's not proper evidence. The dangers of what I call the inferential leap, that these PVCs are directly correlated to death. So it makes common sense that you treat the PVCs and that should lead to less death. But that's not always the case. And a lesson of unchecked assumptions or unknown unknowns. These were the brightest people at the brightest institutions who really cared for these patients, but yet they didn't realize what they were doing was actually causing harm. And this story, I start off with this because part of this actually helped pioneer the evidence-based medicine movement, which I've got a definition there of using the best external evidence to ensure the safety and efficacy of therapeutic regimens. Now, who would agree with that definition? All right, I see some nods, So we've got some good news. That's what I thought too. But the bad news is it's actually wrong, or it's incomplete, not necessarily wrong. And the term evidence-based medicine was originally coined by a man named David Sackett in 1996, which he outlined in a paper in the BMJ. But rather, the scientific evidence is just one part of evidence-based medicine. And the other pieces are clinical judgment to know, does the patient in front of you look like the patients who are in these studies? Are there any unchecked assumptions that might be missing from the evidence? And probably most importantly, patients' values and preferences. Patient-centered care is why we do what we do. And their values and preferences really should be at the forefront. You might have two patients who look exactly the same, where one would be willing to take a tablet every single day to prevent some event, where somebody else wouldn't want to. And that's a big part of evidence-based medicine. And so an easy learning assessment to start. So according to David Sackett, which component constitutes evidence-based medicine? A, the best available evidence. B, clinical expertise. C, patients' values and preferences. Or D, all of the above. All right, we've got lots of D's on the chat and looks like got some nods for D too. All right, so good job, guys. Wait, did it? There we go. I spent like 10 minutes on that animation. Okay, and so now fast forward about 10 to 20 years, and a man, John Ioannidis. So John Ioannidis is a professor of medicine and epidemiology at Stanford, and he's actually the co-director of the Research Innovation Center at Stanford, or rather the Meta Research Innovation Center. And meta research is the research of research. So the man knows research. And one of his papers really caught my attention. And that was a report to David Sackett that evidence-based medicine has been hijacked. And this paper, along with two others, really caught my attention. One of which is actually one of the most highly cited papers in biomedical literature. And that's why most published research findings are false and how to survive the medical misinformation mess. And he pretty much had four takeaways in these papers. One is that much public research is unreliable and offers no benefit to patients. That most healthcare professionals are unaware of this problem and lack the skills to evaluate evidence. That industry-funded trials are often well performed, but often ask the wrong questions, perform the wrong analysis, and have limited transparency in their protocols and data. And that our system is influenced by conflicts of interest and the wrong incentives for research and publication. Whoa. Now, those are some bold claims. And I think a little dramatic, and I don't 100% agree with all of his points, but he does bring up some very valid points. And I'm just going to go through some of the limitations that he discusses today. So starting off with patient representation. So do the patients that we actually see here at Emory and in practice actually look like the ones that we see in clinical trials? And I think most of us know that the answer to that is generally not exactly. But an interesting study that helped to quantify this for me was a literature review that was published in 2015 of articles from 2003 to 2013 that assessed the external validity of RCTs during that time period. And there were kind of two different methods here. One was looking at studies that compared the baseline characteristics of patients in studies versus the RCTs, and a second method of reviewing those who would have met the inclusion-exclusion criteria in randomized controlled trials. And what they saw was about 71% of patients were not representative of clinical practice. And I think that kind of makes sense, right? That usually in trials, patients are assessed for eligibility. And this is generally a carefully selected group. And then those are filtered through, and then they meet inclusion-exclusion criteria. And then they're filtered through again, and then you get your actual trial participants, right? Well, not all the time. And that's where sometimes there is an additional step. And one of those examples are run-in periods. And this is basically a preliminary phase of treatment where only those who meet certain criteria actually enter the trial while they're either being treated or while they're being closely observed. And so a visual representation I've made there. So for example, patients are screened for certain inclusion-exclusion criteria. Then they enter this phase where they receive a study medication, for example. And for those that have adverse effects or have unsatisfactory compliance, whatever is designated as the minimum adherence, then do not get included in the study. And ultimately, those that did meet the criteria are then randomized. And what this leads to is basically an enriched sample. And enriched is basically a fancy word for saying increased response probabilities and increased statistical power for the study. And an example of this is the Paradigm HF study. So this is a very well-known study in heart failure. And this was actually Entresto's pivotal trial that actually pretty much made it now first line in the guidelines for heart failure with reduced ejection fraction. And so it was the drug Entresto, which is sacubitril valsartan, versus enalapril. And they studied the primary endpoint of cardiovascular mortality and heart failure hospitalizations. And if you look on the bottom right, this is what the study assessed. So about 8,400 patients who were randomized to either Entresto 200 milligrams BID, which at the time was called the LCZ 696, which is also now my license plate, and enalapril 10 milligrams BID. But that's not it. There's actually more to it than that. And first, there is a screening period. And then after that, patients underwent a run-in period of enalapril 10 milligrams BID, which is a decent dose of enalapril. And of those carefully selected patients, 10% of those patients were discontinued, primarily due to adverse effects. And then there was another run-in period of a few weeks of the patients who got switched to Entresto 100 milligrams BID, followed by up titration to 200 milligrams BID. And then another 10% of patients discontinued. So at this point, basically 20% of the patients are now gone and aren't randomized. And run-in periods happen pretty often in cardiology. So from 2005 to 2016, about 34% of the studies that led to drug approvals had run-in periods. And in general, for all studies that do have run-in periods, a median of 16% actually end up being excluded. So run-in periods can lead to an underreporting of adverse effects, like hypotension in the case of Entresto. But something I wondered was, are randomized controlled trials even powered to detect differences in adverse effects in general? What was news to me is, or it came to be from this paper that I think was pretty interesting, of the detection of adverse effects in randomized clinical trials. Can we really say new medications are safe? And in general, rare adverse effects, they're usually detected in the post-marketing phase. And that kind of makes sense because you need either lots and lots of patients or multiple clinical trials or a systematic review or meta-analysis to actually be able to detect these rare adverse effects. And it's estimated that anywhere between 20% of new medications will obtain their black box warning through post-marketing. And that in general, most RCTs are not powered in advance to actually detect for most differences in adverse effects. But regardless, statistical testing is still routinely performed. And one study that did a pretty good job at outlining this was a study in 2009 published in the Journal of Clinical Epidemiology. And this was a systematic review of RCTs in high impact journals in 2006. So the high impact journals are the ones you typically know by name, like the JAMAs and the New England journals, who had a primary efficacy outpoint that was positive. And then for their adverse effects, they looked at those that had at least a twofold difference in adverse effects. So those where it's reasonably likely that there's going to be a difference. And of the studies, none of them actually reported what power was needed to calculate the difference in adverse effects, but 3 out of the 6 concluded that there was no difference despite being underpowered to do so. And this is known as a type 2 error, that basically saying no difference exists in these adverse effects, but when in reality, a difference may exist. And next, I'm going to talk about publication bias. So publication bias is, I think, a phenomenon many of us are probably familiar with, of a failure to publish study results based on its direction or strength. So direction being whether or not it's positive or negative, and strength being actually the magnitude of benefit that you see. And I think a unique way of looking at this was from a study from Flacco and colleagues, who looked at a random sample of RCTs that had sample sizes of greater than 100 from 2011. And they ended up with a total of about 319 RCTs. And of the ones that had a non-inferiority design or an equivalence design, 96% of the industry-funded trials had positive findings. So one can make the argument, why even run these studies? I mean, they're kind of wasting their time and resources. Why even do all this? Well, clearly that's not the case, right? This is most likely a matter of either publication bias or some sort of design bias. And 83% of the industry-funded RCTs had favorable results versus 58% if it was non-industry or from a nonprofit group. And another example of publication bias was back to that CAST trial that I mentioned earlier. So remember that the CAST trial was published in 1991, but back in 1980, there was a study of lorcainide versus placebo. And lorcainide is like a cousin of flecainide, basically. And it was a relatively small study of just 100 patients that was powered to see a 25% decrease in PVCs. Now, it wasn't powered for death, but what they did notice was 9 of the 48 died in the lorcainide group versus 1 of the 47 in the placebo group. And the authors did the right thing since it wasn't powered, and they didn't make any bold conclusions, but they did feel like these results were important enough to publish. But unfortunately, their submissions were immediately rejected. They tried, in the story is outlined in the paper in 1993, but basically they tried getting it published in the Lancet. They tried a few cardiology journals, immediately rejected, and they kind of gave up on it until the results of the CAST trial came out. And then they tried again. And at that point, they were able to get it published pretty easily. And the moral of the story that they outline in the paper is that publication bias isn't always just necessarily what the results are, if it's positive or negative, but just what they call the fashion of the day. So whatever interests clinicians or whatever interests the medical journals and reviewers. And because of this problem of publication bias, the FDA was well aware of this. And so in 2007, they passed the FDA Amendments Act. And one portion of this act was that eligible trials, by the time that they have their primary completion date, they've got to publish their results or report their results within 1 year. And DeVito and colleagues in 2020, they did a study to see, well, how well is this actually being adhered to? So they did a cohort study of all registered trials on clinicaltrials.gov for March of 2018 to September of 2019. And the results are on the Kaplan-Meier curve to the right. And this is a Kaplan-Meier curve of the unreported results from the primary completion date. So the x-axis is time since the primary results, and the y-axis is the percent of reports that were actually unreported. And that dotted line in the middle, that's that 1 year mark. And the red line are industry-sponsored studies, and the blue line are non-industry funded studies. And I think what's interesting is at the 1 year mark, the industry-funded studies actually had a higher proportion of studies that were actually reported. So I think these are very encouraging findings. And to me, I think that it's not always malice, that these industry studies, they usually have more research experience, better internal processes, better governance, as opposed to non-industry funded trials sometimes. And then about 1,000 days out, there still remained about 36% of studies that still remain unreported. And next, we're going to go into conflicts of interest. So, conflicts of interest, they didn't really interest me too much until I saw this paper by Steve Nissen. And Steve Nissen, I consider kind of a legend in cardiology. And he holds a lot of hats. He wears a lot of hats. But just to name a few, is that he was the past president of the American College of Cardiology. He was the past chair of the FDA Cardio-Renal Advisory Panel. And he's been on the primary investigator for a lot of landmark trials in cardiology. And in his paper, he outlined a few points that no conceivable logic, in his words, could defend promotional speakers and stockholders on guideline writing committees, and that deliberations of these guidelines take place in secret, and that professional societies forbid disclosing deliberations. So hearing this from him really, caught my eye. And an interesting study that was done that he cited from back in 2002 was a survey. And this was a survey of the authors of 44 different clinical guidelines that were endorsed by North American and European societies. And of the authors that responded, 59% said they had some relationship with the company in the guideline process. And what I thought was funny that 7% believed the relationships influenced their personal recommendations. But of course, when it came to their colleagues, 19% of their recommendations were influenced. And at least in cardiology, through 2008, 17% or 17 of the joint ACC-AHA guidelines, 56% had conflicts of interest. And generally, I don't think this is much of a concern, but what's interesting is when looking at the levels of evidence through the guidelines from 2008 to 2018, what was interesting is what I kind of have labeled here on this table, where the x-axis is the level of evidence. So level of evidence A is from multiple RCTs, and level of evidence C is expert opinion, and B is either based off of observational studies or one RCT. And the x-axis there on top, class 1 recommendations are you should do. Class 2 is basically, perhaps, because there's conflicting evidence. And class 3 is you shouldn't do. And what I just want to highlight is that for class 1 recommendations, so the fairly strong recommendations, the majority of these are not coming from multiple randomized controlled trials. So what's sometimes noted is that they're more likely to be swayed in giving a strong recommendation that's really just based off of either a not strong study or expert opinion. But something that I want to point out that I didn't see too often noted in these papers is that conflicts of interest is not the same thing as proof of bias. I think if I said that, then that would be the inferential leap, right, that we talked about earlier. And I don't think that's the case. And at least the ACC-AHA, they recognize some of the controversy. And in 2010, they actually have now newer restrictions where they limit the conflicts of interest on the writing committee to less than 50%. But what is important is to just be aware that conflicts of interest inevitably exist. I mean, most advancements, especially through cardiology, it's going to come through industry ties. We do have to partner with the industry. But it's important that you shouldn't just take any recommendation just at face value. It's important to critically appraise the guidelines and evidence for yourself and use your clinical judgment. Those are two of the three pillars of evidence-based medicine that I'm talking about. And next, we're going to talk about design bias. And I think I could pretty much sum up design bias by this little cartoon here, which I love, which is one scientist talking to another. And he asks him, how far did you get? And he says, I've got the conclusions. Now I just got to write the protocol. And I think it's it's funny and a little bit accurate. And what I want to get to is the question of, are studies actually asking the right questions or answering the right questions? And that leads to surrogate endpoints. So surrogate endpoints are substitutes for a clinically meaningful endpoint that are typically observed earlier than the endpoints that actually matter to clinicians and patients. So some examples are biologic markers like A1C, blood pressure, cholesterol, or certain clinical events like PVCs, or sometimes even non-fatal MI or cause-specific hospitalizations, some can even argue. And from 2005 to 2012, 45% of FDA approvals were on the basis of surrogate endpoints. In my area of cardiology, we're notorious for this, where 85% of cardiology and diabetes and lipid approvals are based off of surrogate endpoints. And the pros of surrogate endpoints are it leads to a reduced duration, cost, and general complexity of studies. But the downsides are the potential for the inferential leap, that things might make sense from a pathophysiology point, but it doesn't always work out that way. The human body is very, very complex, and potential off-target effects that you just don't know about. Our medications aren't nearly as specific to a certain target or receptor as we'd like to think sometimes. And you're probably wondering, what's wrong with this? Well, I think one story that captures this is a story of the reminder that every rose has its thorn. And the rose in this situation was rosiglitazone. Oh boy, did rosiglitazone get it wrong? And to recap the story, basically in 1999, rosiglitazone was FDA approved on a surrogate marker, which was a decrease in A1C and a decrease in blood glucose. And they actually didn't assess cardiovascular outcomes. And this is a problem because in diabetes, the number one macrovascular complication is cardiovascular disease. And in 2007, Steve Nissen was actually part of a meta-analysis of rosiglitazone, and they saw an increase in myocardial infarctions. So a drug used for diabetes increased the risk of myocardial infarctions. And this actually led to a REMS program for rosiglitazone for a while, which has since been removed. But the good news was the FDA realized that for now, diabetes drugs that are going to come on the market, you have to at least assess cardiovascular outcomes and show that it's not any worse. Not even that it's better, just not any worse. And this is actually how the SGLT2 inhibitors being used in heart failure kind of came about, where they initially noticed that there was a trend towards decreased heart failure hospitalizations. And they took those hypothesis generating findings and studied it in RCT. And rosiglitazone is not the only drug. There are plenty of other drugs that have a similar story, unfortunately. And this is outlined in a 2013 paper of Surrogate Outcomes in Clinical Trials, a Cautionary Tale. And what I have here is a table of drugs that were approved on surrogate endpoints that were ultimately proven harmful. Not even that they weren't effective in the actual outcome they were looking for, but were actually the opposite. And for the sake of time, I won't go through the entire table, but the first two are ones that we've already discussed, so flecainide and rosiglitazone. But I encourage you guys to look into the story of estrogen and progestin in postmenopausal women, and a few other drugs down the line, which you've probably never heard of because they've since been removed from the market. Okay, so now a thought experiment. So if a treatment reduces the risk of heart attacks or myocardial infarctions, would you expect it to reduce all-cause mortality, cardiovascular mortality, both or neither? You don't necessarily have to answer, but it's good to think about it. And this question, it came to my attention when I saw this poll on Twitter from David L. Brown, who's a cardiologist. And on Twitter, he's known as the less is more cardiologist. I think he's got to come up with a better name. But I thought the answer was both. And to me, that makes sense. And that's what the majority of people that answered the question thought as well. And that leads me to the Fourier trial. So the Fourier trial is a very well-known trial to me, and it's a medication in my specialty space. And this was a 27,000 patient study looking at the PCSK9 inhibitor Repatha, also known as evolocumab, versus placebo. And it was studied in patients who had atherosclerotic cardiovascular disease. And all you really have to know about Repatha is that this thing is amazing at decreasing LDL cholesterol. It decreased LDL cholesterol to levels that we had never seen before, about 50 to 60% decrease, and down to about 30s and 40s of LDL cholesterol. So very effective. And in their results, they saw a decrease in myocardial infarctions, or the risk of myocardial infarctions. And I've got it highlighted there in green, so it's easy to see. But the risk was 3.4% in the Repatha group versus 4.6% in the placebo group. But what's interesting is that for cardiovascular death and overall mortality, there was actually a numerical increase in both events. And to me, I thought this was interesting because this wasn't even a trend in the right direction. And we're talking about 27,000 patients. This was a humongous trial where generally, if you're going to see a difference, that difference is going to exist there. And this was interesting to me because for the longest time, myocardial infarctions were a good surrogate marker for overall mortality. And this started back in 1993, where Eugene Brunwald, who's another legendary cardiologist, they advocated to not have the need to just assess all-cause mortality because we couldn't wait that long to get treatments approved. And this was the pre-PCI era or the pre-stent era. And so at that point, because they were so highly correlated, myocardial infarction, cardiovascular death, and mortality, this was kind of the beginning of the composite endpoints in cardiology. But times have changed, and there was a meta-analysis and commentary published on this, where basically they realized that myocardial infarctions are no longer a good surrogate marker for mortality. And there's a few reasons for this. One is that the management today in 2023 is so much better than it was in 1993. We live in a great time in the modern era, but also definitions change. We have a lot more sensitive biomarkers now. We have high sensitivity troponins that can detect MIs that you can never detect before. There's different peri-procedural definitions of MIs, and it's not really clear what a clinically meaning MI is. And studies don't actually differentiate the type of MIs in studies, and their data is not necessarily open for you to take a look. But there was another group that also thought this was very interesting, and that leads me to talk about reproducibility. And so there is a group out there called the REACT group, or the Restoring Invisible and Abandoned Trials group. Now, this is pretty hilarious. On their website, they say that they're trying to fight bias in medicine, and they're kind of investigators on their own. And what they wanted to do was access the data for the Fourier trial and reanalyze it for itself. But getting the data wasn't so easy. And what they outlined in their study is they first had to email the primary investigator, at least on 4 different attempts that were a few months out. They were very passive aggressive and they had read receipts on the email. And they read every single email, but they didn't get back to them to the results. So the next step was going to the FDA. And they reached out to the FDA, but the FDA said it would take 3 to 4 years to address the petition, and then 2 to 4 years after that to actually release the data to them. So they did what a lot of Americans do when they don't get what they want, and they went to Canada, and they went to the Canadian equivalent of the FDA, and they got the results just like that. And what they concluded from their reanalysis was very interesting. And they mentioned that after readjudication, the deaths of cardiac origin were numerically higher in the evolocumab group, suggesting possible cardiac harm. So I'll go ahead and say I don't agree with their conclusion necessarily based on the data, but this was very interesting. And the results of their reanalysis, the main results are here, they're on this table. So it's showing the different causes of death based off of adjudication. And adjudication is basically the process of classifying the clinical event that occurs. So if someone dies of a myocardial infarction, sometimes it's not so clear whether or not they died of the myocardial infarction, or if they were, being treated for sepsis at the time, was it the sepsis? And so you have to have a committee that actually assesses this based on predetermined criteria. And the EVO stands for evolocumab, the RA stands for reanalysis, and the NEJM was the original paper that was published in New England Journal of Medicine. And what they saw was for cardiovascular death, in the original paper, there were 11 more deaths of cardiovascular cause in the evolocumab group. But in their reanalysis, there were 24 more cardiovascular deaths in the evolocumab group. And what was also interesting was that for the 870 total deaths that occurred in the study, 41% of the time, the actual clinical events committee for the study differed from the actual local investigator who was on site. And to me, what this means and what was really interesting, even though their analysis definitely had some limitations, that different authors can come up with different conclusions depending on who's looking at the data. The fact that data is not openly available and not easy to get from these studies, and that endpoint adjudication, it's not as objective, at least how I first saw it. And depending on how it's defined and depending on the criteria that you use, you can get pretty different results. And so we talked about reproducibility from the standpoint of reproducing the same data, but how about reproducing data at a later time? Or the way I think about it is, does evidence have an expiration date? One of the many things that differentiates a case report from a randomized trial is that you should be able to replicate this in patients down the line once the results actually come out. And this is actually why the FDA typically requires 2 or 3 studies for an FDA indication. And in the study in 2005, John Unides' group, they looked at studies published between 1990 and 2003 in high impact journals that were highly cited. So at least 1,000 citations. And of the studies he looked at, 92% were what they defined as effective studies. And effective studies he defined as basically in the conclusions if they determine that the benefit outweighed the risk. And of those 45 studies, 44% were able to be replicated later on in the future, but 24% still remained unchallenged. 16% were actually contradicted, so went in the other direction, and then 16% of the studies ended up having a weaker effects in subsequent studies. And so my takeaway from this is that we should probably be cautious about drawing conclusions from just one paper, and that sometimes clinical endpoint adjudication is not as objective as it may seem. And I encourage all of us, and myself included, to check out how these definitions change over time, really critically think about whether or not this is an appropriate clinical endpoint. Okay, and so now a learning assessment. So the REATS group reanalysis of the 4-year trial was entirely consistent with the findings of the original 4-year trial. True or false? You guys can say out loud. False, be confident. I like it, yes. False is correct. And now I got, yep, the chat, they've got it as well. Okay, and last we're going to talk about some of the difficulties in the interpretation of data. So I'm going to start off with what's known as the RCT paradox. So the way that we currently do things is that we figure out whether an intervention works based on population data, based on large samples of people, and we study them and assess the endpoint that we're looking for. But then we're supposed to take that population data and apply it to a single patient. But what's difficult is that you'll never actually know whether or not the individual patient is going to achieve the benefit that was seen in RCTs. And the paradox is that clinical trials are the best way to figure out whether or not an intervention works, but it's the worst way to figure out who will actually benefit from it. And so one way to think about this is to imagine an intervention increases the life expectancy of 10 patients by an average of 1 year. Well, there's 2 ways of kind of looking at this, which Daryl Francis and his group kind of show us here, which is in green, perhaps everyone gets 1 year of benefit. But in orange, in the other scenario, one of those 10 patients can gain 10 years while the rest gain nothing at all. And so in order to kind of provide a visual of this, they did something pretty clever. And to start, I want you guys to know that based on a Cochrane review, statins reduce cardiovascular death by 20 to 30%. And that's a relative risk reduction over the course of 5 to 10 years. But we ask patients to take statins specifically for primary prevention for the rest of their life, for 20 or 30 years. And so I want you guys to think that if we were to ask 100 patients to take a statin every day for 30 years for primary prevention, so those who haven't yet had like a heart attack or a stroke, would they all have a better health outcome because of it? Even if it's 1, 2, 3 months worth of living longer lifespan. And so the way they try to figure this out was in this paper called The Distribution of Lifespan Gain from Primary Prevention Intervention. And this was basically a Monte Carlo simulation. And in their simulation, what they added in was the UK cardiovascular and non-cardiovascular mortality data, the relative risk reduction of 20-30% in cardiovascular death, and the fact that non-cardiovascular death progressively rises with age that won't always be reduced by statins. As we get older, our risk of dying from falls, pneumonia, COPD, all of those go up, where statins aren't necessarily going to reduce that risk. And just based off probability alone, people die of car accidents, for example, and you can't necessarily account for that unless you have a really, really long-term study. And what they found was that the lifespan gains, they're actually concentrated in an unpredictable minority. And this blew my mind, and I never would have thought of primary prevention statins in this way. And the fact that most actually get no benefit at all, but some get a major benefit. And so the results are kind of shown there and outlined on the right, where each one of those little squares is like an individual patient. And in those that's gray basically get no additional lifespan at all. But then a small minority of patients in green get a pretty large benefit. So in this scenario, 7% of patients get an average of 99 months. So a huge gain, but 93% don't really get anything at all, even though the actual mean gain or the average gain was about 7 months. And what I thought was even more interesting is that younger people who are at lower risk, they have a lower probability of gain, but they actually achieve a higher gain. And so a way to think about this is if you prevented a fatal MI in someone who is 51 years old, that's pretty rare for that to occur. But if it does occur, then you've got many more years to go. But if you have an older, higher risk adult, which is traditionally who would we treat with statins, they have a higher probability of gain, but they have less gain because now they have more competing causes of death that the statin won't actually benefit. And so this really blew my mind. And my takeaway from this is when counseling patients, we should find a way to try and actually communicate to them. And it's important to have a shared patient understanding of the different benefits and risk, understanding your individual patient's baseline risk, and ensuring that the decision actually aligns with their values and preferences. And you shouldn't really necessarily care whether or not a patient decides to take a medication or not, but you should care whether or not they've been properly informed of the potential benefits and risks. And so, you should ask yourself, do you buy lottery tickets? Because that's kind of how some of these interventions actually work. All right, so I have purposely only gone through the surface level of some of the difficulties and limitations of evidence-based medicine, the modern movement. And I encourage you guys to look deeper into this. And I'll leave you guys with a few quotes. So one of them is that it is simply no longer possible to believe much of the clinical research that is published or to rely on the judgment of trusted physicians or authoritative medical guidelines. I take no pleasure in this conclusion, which I reached slowly and reluctantly over my 2 decades as editor of the New England Journal of Medicine. So this came from Marcia Angel, and she was actually the first woman editor-in-chief of the New England Journal of Medicine. And that medical journals are an extension of the marketing arm of pharmaceutical companies. This came from Richard Smith, who is the former editor of the BMJ, and he outlines a lot of his points in his essay in PLOS Medicine, in that journals have devolved into information laundering operations for the pharmaceutical industry. And this is from Richard Horton, who's the current editor-in-chief of The Lancet. All right, so where do we go from here? So I remain very optimistic, okay? This is how science progresses This is how anything progresses. You do have to acknowledge just that there are some natural limitations and uncertainty. And I think that's okay. The evidence-based medicine and medical evidence, this is invaluable to patient care. I don't think that can be understated, but it's not infallible. Your residents, I know you're probably getting excited, but you still have to learn the class 1 recommendations and the guidelines. You've got to know the main results of these trials. But then I encourage you to push further. You've got to crawl before you can walk, before you can ultimately run. And it's important, I think, to have a sense of humility, I think, to what we're doing, and that there is a lot of uncertainty. And I want you to think back to the CAST trial of the smartest people at the smartest institutions who meant well. They didn't know that they were even causing harm. And that there's huge uncertainty in healthcare, but that comes with the territory. I think you just focus on what you can control, you assume things work, and you communicate that to patients. And I think the little things matter more than ever. And when I think of the little things, I think about communicating to patients, making sure they feel comfortable, doing a proper med rec, clarifying allergies. I put so much more effort into that stuff now, whether or not the individual P2Y12 inhibitor a patient gets. And I think it's good to be a little bit of a health skeptic or a healthy skeptic. And this is not easy. I do think there's sometimes a bit of even FOMO in this, where you kind of want to be on board with what's exciting, what's the general consensus, and it's hard to go against the grain on that. And that's something that I am working on, but I think is important to have for patient care, and the importance of shared understanding. In understanding that there will always be the existence of gray zones and benefits fall in a continuum. And what I've realized that ultimately the right decision ends up being on whatever the patient's preference really is. And one way to do this is through patient decision aids. So I haven't used these personally, but there's a lot of data out there on the benefits of patient decision aids, on kind of outlining the benefits and harms that are seen in studies, and kind of giving that to the patient and having them think about it and make a decision. And working on communication and communication of risk. This is not an easy thing to do. Humans are naturally bad at perceiving risk and communicating risk. So it's something that I am continuously working on. And I think there's a great campaign out there. It's called the Choosing Wisely campaign. I send patients to this website very often, and I encourage you guys to check it out. But this campaign is all about sparking conversations between clinicians and patients to determine if treatments are actually needed, what will happen if I don't do this treatment, what will happen if I do this treatment. And working on critical appraisal and medical evidence. I still do believe that the best evidence is the most available evidence, regardless of publication bias. And I think it's important to work on our basic critical appraisal skills. This is something that's not easy, but it's something that you should work towards getting better year after year in order to provide the best patient care. And realize that there's a lot of nuance in decisions and interventions, and avoiding what David Sackett called cookbook medicine. I think it's not enough to know that a treatment is beneficial or that it has a benefit. I think you should know generally what is the benefit, how much of a benefit, who exactly will benefit. I think those are things that helped me kind of try and get to the next level. And I've got to give a shout out to Colin's course. It's a great course on critical appraisal. And I think it's a great place to start. And there are many ongoing initiatives. For the sake of time, I won't go through all of these, but I really like JAMA's series of Less is More, where they highlight some of the difficulties of overtreatment and overuse in medicine. And BMJ's evidence-based medicine is also a fantastic resource. All right, and I'd like to acknowledge one of my good friends and old classmates, Divian Nguyen, for helping me with this presentation, and Michelle Morales, one of my ambulatory care colleagues. I'd like to thank you as well for all your help. And I'd also like to give credit to all of these very smart people here. I have not met any of them, but their work has been stuff that I've been following for the last few years, and it's really opened my eyes, and I think has made me a better clinician. And I highly recommend everyone read the book, Ending Medical Reversal by Vinay Prasad and Adam Sifu, for unfortunately many stories just like the CAST trial. And with that, I will take any questions.